Thank you very much for the invitation to present our first results about this topic. As had been said, I'm going to talk about the possible detection of peptides in sediments in Rio Tinto. And the overview of my talk is divided into a big introduction. I hope not to be very repetitive. And I'm going to show you our preliminary results is preliminary and a brief summary. As you know, organic matter is present in all the environments, in soils, in the water. It is present in general, in every place on our planet. And this organic matter is derived from cells or the metabolic activity of many of the organisms, from plants, animals, fungi, bacteria, etc. The important thing is that all the cells are composed by the same kind of molecules. And this is important because the functions like the storage, the structure, the transfer of information are played by polymers such as nucleic acids, the lipids, proteins, or polysaccharides. When the organism dies, the molecules that form it start a long rate of degradation. But degradation are not the same for the different compounds that form the organism. The nature and the structure of the molecule influences the pathways, but also the environment. In paleontology, we can say that the molecules that bring us clues about the past or that can be related with past life, with biological activity, are called biomarkers. But the thing is that some molecules cannot be undoubtedly considered as biomarkers because they can also be produced in simulation experiments, or they can be detected on extraterrestrial bodies such as meteorites or comets. So this is the case of the most evident molecules of biomolecules like amino acids, sugars, and carboxylic acids. They have all these, both of these possibilities. They can also be produced abiotically or can be detected in bodies like meteorites. But it doesn't happen with polymers. Polymers are so complex that are undoubtedly confused. So proteins, for example, are very important for organisms, for living beings, because they function as structure, catalysis, movement, the regulation, and transport. But the problem with proteins as biomarkers is that they are very labile. So they are lost during fossilization because the hydrolysis of the peptide bond. However, we are very interested in that issue because the determination of the molecular and the degree of preservation of organic matter in extreme conditions is a challenge in molecular paleobiology and also in astrobiological studies. So our premise was to determine, if it is possible to determine at first, the presence of peptides or residues, oligopeptides, in the sediments of Rio Tinto that are exposed all the time to extreme conditions. And Rio Tinto, as you know, is a Martian analog. It's considered a Martian analog. And it is located in Spain, in the south part of the country, close to Nerva, that is a downtown, a small downtown. And it's very unique because it has a flow with red water, and it is caused by high content of heavy metals, and also the pH is very acid, but very acid is close to 2. And it's characterized by an important microbial community. I want to show you the water of the Rio Tinto. Our results were based where we took 5 different samples. One is modern, the modern deposit, and 4 were ancient, and they range from 5,000 to 7 million years. And you can see here the points of sampling. The first thing that we wanted to know is to corroborate if we could have possibilities to find peptides or not. So we made an FTIR spectroscopy, just for having a clue if it was possible to detect the carboxyl group. And you can see here that in the young terras, the intermediate terras, and in the old terras, we can find this vibration that is characteristic of amide. So we continue having this as a starting point, and we follow a protocol for the extraction of some peptides and nucleic acids. It's a protocol that has been published, not for us. And we, at the end, we recuperated an extract and we weighted it. As you can see, the amount are very small. This is 2.82 milligrams per gram of the sample. It's very, but very little. And it is a decrease in the amount of the extract in function of time. This is the younger, and this is the oldest sample. With this extract, we made dilutions, so we wanted to follow traditional determination of protocols, determination of proteins with colorimetric assays, but it was not possible because even if we diluted very small amounts of the protein, in this case was a protein, a non-protein to make the calibration curve, our results were very close to this area, so we considered this was not a good method for the determination and quantification of proteins, but just in this case. So we had the opportunity to use another technique that is called Fourier transform ion cyclotron resonance mass spectrometry that allowed us to have ultra-high resolution mass spectra in Germany. And I want to show you just that this is just an example of the spectra that we can obtain with this equipment. And you can see the ions that we produce. But the important thing is that we could identify 5 different peaks with a weight more than 1,000. In this case, it's 1,053, the example that I'm showing you. And you can see this is the young terrace, and the intensity is very high. In this case, in blue, is the intermediate terrace, and the smallest is the old terrace. So as you can see, this peak diminished with time. We are continuing doing the experiments to try to identify the nature of course, we think it is a polymer. We suppose it is a peptide, but we are continuing our experiments to verify it. It is also the other peak that we analyze. As I said, we could find 5 different peaks. And as a summary, I want to say that according to these preliminary results, I really emphasize it because we are still working with the samples, and it's very risky to say that we can find proteins, but we think that we could, all these results suggest us the presence of oligopeptides because of the weight in the sediments of Rio Tinto. We tried different methods. Of course, we could use any others, and we are thinking about this, about doing this. And these results suggest that the conservation of small and short peptides is possible even in the adverse environments like the acid conditions, and high metal concentration. And in future work, we want to continue analyzing the samples that I didn't present you, that are the recent sample, that is the youngest, and the oldest one, that is called Gossin, and to study the decomposition of peptides under the conditions that we know Rio Tinto has. For example, very low pHs, and heavy metal concentration to study the dynamic of the composition of the proteins. And I think, yes, it is all. Thank you for your attention.
Thank you. We still have a few minutes for questions.